This is the instant blood glucose meter, and this is how you're supposed to interpret your blood glucose reading. This table of interpretation that's showing some intervals was derived from a specific healthy population, or it is known as the normal ranges or the reference intervals. Then, since these intervals are obtained from a specific and huge number of population, then it should only be suitable for most people in that population, but not all of them. Because we know that each of us is different and has unique biological and genetic characteristics. Then we may ask, would it be possible to have our own reference intervals? My name is Murih Pusparum, and to answer that question, I would like to present our research with title From Population to Subject-Specific Reference Intervals. This research is in collaboration between Hasselt University, Belgium, and Flemish Institute for Technological Research, or VITO. This presentation will be divided into 3 sections. First, I will explain about the concept of population reference intervals. Second, I will explain about the methodology. And third, we will discuss the results of the subject-specific reference intervals. For the definition of population reference intervals, or PRI, I will give an example from the HDL cholesterol measurement. So in this case, the PRIs are calculated from HDL levels of a healthy individual population. The boundaries of the interval are usually set to 2.5% and 97.5%, so that we will have 95% intervals of normal HDL level. This interval can be used for interpreting the lab test results of HDL of a patient. So when this result falls outside the intervals, then the doctor or the clinician will examine the patient further. However, these PRIs have some limitations, such as it requires a large cross-sectional dataset, or a dataset that only contains one measurement per subject at one time, and it is also assumed to be constant over time and space. Moreover, a Gaussian distribution is assumed especially in the parametric estimation method. The methodology includes the explanation of the data and statistical methods. This research uses 2 types of datasets. The first is the Balearic data, which comes from a study in the Balearic Islands, Spain, and this is a cross-sectional dataset that was only measured at one time point. It consists of several clinical and physiological measurements of 60,000 participants with age ranges from 19 to 70 years old. The second is a unique longitudinal dataset of 30 participants collected by VITO. This dataset also includes physiological and monthly clinical lab tests that collected over a 9-month period. The age of the participants ranges from 45 to 60. For this research, we only include 4 target parameters. They are glucose level, HDL cholesterol, systolic blood pressure, and waist circumference. First, we employed the classical parametric and non-parametric reference intervals estimation methods to the cross-sectional data of the Balearic Island study. Here in the parametric method, it uses the 97.5 percentile of standard normal distribution. Then we also applied the non-parametric method using the ordered set of sample observations. 2 non-parametric approaches were used in combination with the bootstrap samples. For the IM frontier longitudinal data, we fitted the linear quantile mixed models in order to derive the 2 bounds of the subject-specific reference intervals, or SSRI. The model includes 1 fixed intercept, specific at a certain level of quantile tau, and 1 random effect. The sum of this fixed intercept and this random effect corresponds to the SSRI for subject I, in which when tau is equal to 0.025, then we will have the lower bound of SSRI, and when tau is equal to 0.975, the upper bound of SSRI is obtained. This figure shows the population reference intervals of Balearic data obtained from the parametric and non-parametric methods. We also compared the result with the published reference intervals, and we can see that our results are wider than the published PRI. Moreover, the bounds are different between males and females. For this figure, it shows the individual profiles of IM frontier data of the 4 target parameters over a 9-month period. For all parameters, we can see that values measured within the same individual are more similar to each other than of different individuals. In other words, a large between subject variability and a small within subject variability were observed. This figure shows the results of SSRIs estimated by LQMM. The x-axis corresponds to the 30 participants, and now finally we can see that each individual has different interval. The horizontal dashed lines are the PRI, so if we compare them, then we can see that some of the individuals have lower 2.5% bound, and some of them also have higher 97.5% bound than the PRIs. That is expected because again every individual has different characteristics, so the reference intervals would also be different. For HDL cholesterol and waist circumference, only one of the 2 bounds were estimated because, biologically speaking, the healthy status threshold can just be defined by either the lower or the upper bound. So we believe that when the SSRIs are widely used in clinical practice, they will allow for more precise diagnosis and hence they will be beneficial both for the patients and clinicians, although more research is still needed to evaluate the LQMM methods to give reliable SSRIs.